Welcome to this webinar organized by EURO-NMD in collaboration with with ERN, Rare Neurological Disorders, EpiCare for Rare Epilepsies, and the European Academy of Neurology. As you know, we are celebrating the Mitochondrial Diseases Awareness Week, and we have organized during this month of September between ourselves webinars on the thematics of mitochondrial disorders. We have today with us Costanza Lamperti, that works as a neurologist in Milan and C.Besta, and she has a PhD on alpha-dystroglycan glycosylation defect in brain and muscle in patients with congenital muscular dystrophies from morphology to molecular genetics. Costanza is an expert on both sides of neurology and genetics, and that's a very fortunate and appropriate cross matrix to develop a good exposition about these themes, and she's rather committed to them for a long time, with some training of happening in the States in Columbia University and some of the training in Italy. She is the coordinator of several studies, a partner of the Italian Register for Mitochondrial Disorders, and many other networks, namely eRare and others. So thank you, Costanza, for being so kind to accept and give us this talk about mitochondrial genetics. The floor is yours. Thank you.
Okay, so hello to everybody. Can you hear me?
Yes, perfect.
Okay, so hello to everybody. I'm very glad to be here to speak about the genetic of mitochondrial disease, that actually is my first field, and in this week, that is the week for the mitochondrial disease. So I'm very happy that we can share this presentation, and I hope that can be helpful for some of you. So my field is mitochondrial disease, and so the protagonist of my talk is the mitochondria. Mitochondria is a small organelle that is the powerhouse of our cells. It supplies the cell energy, supplies the cell with the energy by the OXPHOS, and is an important is the host of most of the important metabolic pathway, like circle of Krebs, beta oxidation, lipid, and cholesterol synthesis. It's important for the homeostasis of calcium and for the apoptotic signal, and it's involved even in the autophagic process. So we can say that the mitochondria are the kind of motor of our cells and producing energy and oxygen. And as what happened when the motor is broken, what happens is that if the motor is broken, we can have an increase of toxics, like in this case, lactic acid and free oxygen radicals, and a decrease of energy because the motor is not working, and oxygen. And this produces the cell death. Each time that we have an alteration of this motor, we can have a mitochondrial disease. So the mitochondrial disease at the end are caused by the incorrect functional of oxidative phosphorylation. As mitochondria are present in all the organs and in all the cells, the mitochondrial disease can involve all organs, like mostly the organs that need more energy, like CNS, muscle, cardiac system, endocrine system. And another characteristic is that they can present in a very variable clinical phenotyping, and ranging from the early stage of life with often more severe phenotype to the very old age of life. But mitochondrial disorder are quite frequent, being anyway rare disease. If we take all the mitochondrial disorder together, we can say that is one of the most frequent metabolic disorder, and its prevalence is estimated in 8,500 births. So again, we can assume that mitochondrial disease can be present at any age with very different phenotype, with any symptoms ranging from floppy baby to just optic alterations, and any course being very severe or being quite benign. But the most important, I can say, and interesting things of mitochondria is that it's the only organ that has a dual genomic control. So we can have mitochondria, and the base of mitochondria is the respiratory change. That's formed from several complexes, 5 complexes, and this complex and his functionality can be regulated by genes that are coming from mtDNA that is present in every cells, and especially for complex 1, 3, 4, and 5, and from DNA, genomic DNA, so that is in the nucleus, that encode for proteins, again, for such of this complex. But the nuclear genome has even an important role in the regulation of mitochondrial DNA by himself, especially in the replication or reparation of mitochondrial DNA. So at the end, we can say that mitochondrial disease can be hereditary in every kind of way. So in a Mendelian way, with disease that are hereditary in autosomical dominant or recessive disease, and X-linked disease, or can be hereditary in a specific way that is a specific way of mitochondrial DNA that is matrilinear. And on this aspect, we go deeper now. So human mtDNA is a molecule of 60,569 base pair. It codifies for 30 structural genes and 25 synthetic genes, and has some characteristics that are typical for mitochondrial DNAs, that is the asynchronous replication, polycystronine transcription, in situ translation, and more important for clinicians is the maternal inheritance and the polyplasm. So what are the rules of mitochondrial DNA? Are for sure, again, the maternal inheritance, the heteroplasmy, the mitotic random, and the threshold effect. And what we mean with this. First of all, each human has thousands of mitochondria, and in each single mitochondria, we have multiple copies of mtDNA. This means that we have a lot of mitochondria and really a lot of mitochondrial DNA here. Usually the mitochondrial DNA is always the same. And so we can say, like here, that we have an homoplasmic mitochondria cells with all the same mitochondrial DNA. But in some cases, mitochondrial DNAs can be mutated, can present some variants that can be pathological or not. In this case, we can have different kind of mitochondria. And in the same cells can coexist the wild-type mitochondrial, so the mitochondrial that are not mutated, and the mitochondrial with the variants of the mutation, so the disease mitochondria. And this phenomenon that is typical for those for mitochondria is called heteroplasmy. So the presence of the two mitochondrial DNAs, the health and the disease one, is the heteroplasmy. And this is another important concept. We know that heteroplasmy the percentage of heteroplasmy can lead to the disease, what means that we can have cells in which we have very low mutant mtDNA or very high mutant DNA. And the percentage or the amount of mutant DNA in the respect of the non-mutant or wild-type DNA is what gives the clinical phenotype and this specific cell or organs affect. And there is a threshold, so a specific amount of percentage of mutant versus wild type that can make the difference in pathogenic phenotype or not pathogenic phenotype. And these thresholds, it's specific for every mitochondrial mutations. And the last, but not least important rules of the mitochondrial DNA is linked to the transmission of the disease. We know that mitochondria, as we said, are present in every cells, and in particular, they are obviously present in the oocyte and the sperm cells. And in the oocyte of a person, we can again have a certain amount of mutant DNA and wild type DNA. During the maturation of the oocyte, there is the formation of primary oocyte, and in this oocyte we have a restriction of the number of mitochondria. So it can happen that few mitochondria go in every oocyte, and it's actually impossible to predict how many mutated mitochondrial DNA will go in one oocyte or in the other. And afterward, in the going on to the maturation and to the mature oocyte, again, there is an expansion of mitochondria. And basing of the amount of mitochondria that we have in this restriction, then we can have a different percentage of mitochondrial DNA mutated versus wild type in the different oocyte. And again, during the fecundation, there is no contribution of mitochondria by cell sperm, and we can so see that in the base of how many mitochondrial mutators are in the mature oocyte, we can have a newborn that can be severe affected if the percentage is very high and the threshold is upper of the ones needed to have the disease, a child with a mild disease or an unaffected disease. And this is absolutely not possible to prevent. We say that there are again two genomes, so we can classify the OXPHOS disease in a defect of the mitochondrial DNA or defect in the nuclear DNA. And for the defect of mitochondrial DNA, again, we can have defect on the protein synthesis of the protein encoding for OXPHOS subunits or large deletions. And this is important for the concept of heteroplasmy or homoplasmy. And then we can have mutation in several genes in the nuclear DNA that are codefined for the different proteins and are involved in the different persons that we'll see later that are present in the mitochondrial functions. So if we speak about mitochondrial DNA, we can see that nowadays we have a lot of described mutation as pathogenic mutations that can be present in every kind of chains and in tRNAs. And another characteristic of this disease that is very interesting and peculiar is that one phenotype, like the Milan's phenotype, can be linked to several mutations in the same gene, like the tRNA of the lysine, but even in mutation in different genes. So one phenotype can be driven for different mutation in different genes, and the different genes can give just one phenotype. The first DNA mutation was described in 1988, but afterwards a lot of mtDNA mutation was described. And there are some mutations that are more frequent in different genes, both in, genes involved in the complex, like ND genes, or in the tRNA. And the most common mutation give specific syndromes of this disease. And the most common are LHON, that is a subacute bilateral consecutive optic atrophy, and Milan's, that is a myopathy with epileptic lactic acidosis and stroke-like episodes. The MERF, that is a myopathy with muscle aggregate fiber and myoclonal epilepsy. The NARP, that is a neuropathy with retinitis pigmentosa and ataxia. And the MILS, that is mitochondrial Lee syndrome. The most common disease linked to a point mutation in mitochondrial DNA, it's the Milan, that is characterized by the early onset of stroke-like episode, deafness, diabetes, epilepsy, and can be ataxia and myopathy. But we can have here different phenotype again. So the same mutation that is 3243 mutation can give a milder phenotype that is that present only with hearing loss, diabetes, and myopathy. And this disease is linked to this mostly to this mutation that is 243 that is in the tRNA of the leucine tRNA, and it's I can say one of the most frequent mitochondrial disease in adults. Another well-known disease, less frequent, is the MRF, that is characterized again by myoclonal epilepsy, ataxia, rugged red fibrous, and can be present frequently lipomas. It's again a mutation in a tRNA, that is the lysine tRNA, and it can, even in this case, can be different phenotype with a predominantly ataxia phenotype or a predominantly myopathic phenotype. As we say together before, the heteroplasmic mutation is can be transmitted, and in this point mutation like Milas, MRF, NARP, etc., we have often an heteroplasmic mother that can be affected or not, dependent of the amount of mutation that he has in his tissues. But their ovocyte can present with different amount of mutation, and because of that mechanism of bottleneck, again we can have the reduction and then the re-expansion of mitochondria, and so having a different presence of wild-type versus mutated mitochondria, and give a rise of healthy, sick, or very sick kids. A particular cases is of mitochondrial disease linked to mtDNA mutation. Point mutations is the LHON. LHON is a disease characterized by the only organ involvement that is the nerve, optic nerve. So there is an optic atrophy that is present and is linked to three more frequent mutations that are always present or as homoplasmic. So the mutation is present as homoplasmic in all tissues, but just the optic nerve is involved. And in this case, the transmission can be different. No. Can be different, sorry, I don't know how to come back. Anyway, can be different because in this case, all the kids have a homoplasmic mutation, but just the male can show the disease. And then we have the alteration of the quantitative or qualitative mtDNA. So not anymore a single mutation, but the presence of large lesions that is called macrodeletions. So we have the coexistence of wild-type DNA and deleted DNA that can give rise of a disease that is called Carcine syndromes. We can have multiple deletions, so different deletion of different dimensions of mtDNA, or that can give a different phenotype that is a progressive external ophthalmoplegia or PO or SANDO. And then we can have a depletion of mtDNA that can present with a Alpert syndrome. The Kearns-Sayre syndrome The Kearns-Sayre syndrome is linked to a depletion of mtDNA and is characterized by an early onset muscle disease, a cardiomyopathy, often linked to an arrhythmic cardiomyopathy, leukoencephalopathy, epilepsy, and short status. And is again a different way of transmission. This is the only forms of mitochondrial disease that is completely sporadic, because all primary oocytes with the mutation don't go on in the maturation, and so all the kids are healthy. Then we can as I mentioned it before, so we have the mtDNA mutation, but the nuclear genes are more in the year most important for the mitochondrial DNA genetics. And again, here we have a lot of function that can be arised from nuclear DNA mutation, and the nuclear genes can codify for different structure and function of mitochondria. One of these structures is the genes that are involved in the protein that are present in the respiratory chains complex. So the structural proteins, a mutation in one of the structural proteins of complex 1, 2, 3, 4, 5 can lead to a mitochondrial disease. We know that the nuclear DNA actually is codify for the most of the protein present in all these complexes. So the prevalence of these proteins in respect of the mtDNA mutation are really high. A second group of disease that can be linked to the genes that can link to the disease of mitochondrial DNA's nuclear genes are the genes that are involved in the assembly genes of the complex. So the complex are present and are constituted by the proteins of the complex and some other proteins that are important to take all the complex and all the proteins of the different proteins of the complex together, and that can make the complex work in the right way. And these are the assembly complex proteins for the complex. And there are a lot of mitochondrial disease linked to the mutation in this specific assembly complex for the complex 1, 2, 3, and etc. And more rare diseases are linked to the lipid biosynthesis, as mentioned before. In mitochondria is really important even in his role in the lipidic and circular Krebs metabolism. So there can be mutations that are mostly present in proteins and enzymes that are present in the metabolic way of lipid. One is, for example, the TATS or tafazine, that is an acyl coenzyme A synthetase translocase. Mutation in these genes can produce a very severe heart disease in kids that it's called Barth syndrome. But one of the most important and common nuclear genes involved in the mitochondrial disease are the genes that are involved in the maintenance and repairs of mitochondrial DNA. There is a repairing maintenance machine that is all codified by the nucleus, and then that can produce a secondary alteration in the mitochondrial DNA. So we can have the multiple deletion, as we say before, and depletion. Both these syndrome are linked to a nuclear gene alteration. And as you can see, the gene can be the same, and the phenotype can change if the gene involved is producing a multiple deletion or a depletion. So the same gene, for example, twinkle, can give a hepatocerebral disease in a child with depletion, just an autosomical dominant PO in adult with multiple deletion. And the same is, for example, for DJK, that again in adult can give a autosomic recessive PO, and in kids can give a severe hepatocerebral disease. Another group of diseases that are now became more frequent and well diagnostic is the genes involved in the tRNA and in the protein apparatus, producing apparatus. So, for example, the amino tRNA synthetase, and the mutation can give a difficulty to the mitochondrial protein to be constructed and a secondary disease in these fields. And last but not least, are the genes involved in the mitochondrial dynamics. We know that mitochondria are not a fixed organelle. If we go to see mitochondrial in vivo, we can see that they are a kind of reticulus, and they are produced by a fusion and fission of mitochondria. And this circulating and fusion or fission of mitochondria is lead to mutation in different gene. And one of the most frequent gene associated with disease one of the well-known gene is OPA1. OPA1 is a gene that is associated with an autosomal-dominant optic atrophy, or ADOA, that gives a visual disorder in child and adult. There is a childhood onset of the disease. The disease usually starts before 10 years of age. And there is an extreme variability in the clinical presentation. There can be child that can be completely blind, or adults that have in the same family, adults that can have just a slow impairment in visual acuity. And again, depending on where the mutation is present, we can have a very severe phenotype that is present in up of 20% of the OPA1 mutated patients. And it's present for example, in the GTPase domain, and it gives a severe phenotype in which we have optic atrophy, but even deafness, severe ataxia, ophthalmoplegia, and peripheral neuropathy. So again, as told before, the mitochondria are linked to the fact in the mitochondrial DNA and the nuclear DNA. And for the diagnostic point of view, we always had, since a few years ago, a kind of strategy that was linked to the for mitochondrial DNA to do the Sanger sequencing of the whole mitochondrial DNA to identify the single mutation, the point mutation, or to do the Southern blot to identify the macrodeletion and the multiple deletion. And for nuclear DNA, what we was used to do was going with Sanger sequencing gene by gene, basing on the biochemistry phenotype on the clinical phenotype. This strategy was very good, and we used this for a long time, but actually we lost a lot of mutation and was very time consuming, and you need a lot of time to have a final diagnosis on those diseases. So in the last years, we had a real improve in the possibility of diagnosis with the new technologies, and in particular with the NGS. The NGS technologies is a technology that allowed to sequence in the same moment a huge number of genes, and actually can be used as target gene panels where specific gene associated with mitochondrial disease can be studied, and there can be around 300 already known genes associated with the disease that can be sequenced at the same moment. The Mito exome, in which we can have 1,000 to 1,500 genes, and there are all genes linked to mitochondrial surviving functionings, but not only the ones that are linked to a disease, but all the mitochondrial genes. And then we can be the clinical exome that is going to sequence 3,000 genes together, and it's genes that are linked to any kind of disease. And at the end, we can have the whole exome sequencing, that is a way in which we are sequencing all the codifying genes, all the codifying genes of the exome. And this is obviously used in nuclear genes, but in the last time we are starting to use it even for mitochondrial mtDNA analysis. So this high these new technologies like next generation sequencing allowed to have a diagnosis even in mitochondrial disease that can be more sophisticated. We can capture more even low present of low amount of mutation, and we can have a better way to see the amount of heteroplasmy, and it's possible even to see the presence of macrodeletion and to quantify them. For DNA, for nuclear DNA, again, even here we have a important progress of the new technologies that we can use, and we pass by again, as I told before, the target panel sequencing that are going to sequence up to 300 or 400 genes, to the whole exome sequencing that again is going to sequence all the exome or the whole genome sequencing, that is to sequence all the genome. So not only the codifying part of the genome, but even the intronic part. All of this methodology have some aspects that are good and some that are less good. The good aspect, for example, for the target sequencing is that it's more rapid and can be is less expensive, but in the other aspect, we can lose some mutations, and we can't lose some new genes, any kind of mutation in new genes not already described. The whole exome sequencer is for sure more better to go to see mutation in genes that are not already described as associated of disease, but is for sure slower, and so for the diagnosis purpose can be not so good at the end, because we can have a very late results. And again, for the whole genome sequencing is again slow, and the other aspect is that we can have a very huge amount of variants, and it can be difficult to analyze all those variants and to understand which variants is really pathogenic and which variants is not pathogenic. But what we can see here is that with the presence of these new technologies, we can have a very increasing of new gene discoveries. And as we can see in this graphic here, we can see that in the beginning, most of the diagnosis and the new genes described was found with mtDNA sequencing or candidate gene by sequencing. But after a certain point, it is around 2010, when these new technologies come out, the screening and the diagnosis with, again, mitochondrial DNA sequencing or candidate genes goes down, and the new technology, like NGS and actually WES, so whole exome sequencing increase and allow to find the most of the new genes that we can describe now. Still, if we analyze the other, not the diagnostic cases, and we go deeper, and we see that still using the WES, so the whole exome sequencing and even the whole genome sequencing, still 50 of the unsolved cases remains unsolved. And or there are some variants that are called VUS, so variants of unspecified significance that we don't know if they are really causing the disease or not causing the disease. And so still we have a lot of work, as looks like, to do, because we can't really say that we are able to diagnose mostly of the mitochondrial DNA, mitochondrial disease. But with this new technology, as you can see, we found really a huge amount of new genes and associated with mitochondrial disease. Some are really rare. I can say there can be genes that are present just in 1 or 2 family in the world, so super rare disease. Otherwise, others can be more frequent. And here I just wanted to give you 2 experience of our lab that can show you how the combining of these technologies can help in find a diagnosis. This first case is a 20-year-old woman. She comes in our outpatients because of a severe cerebellar ataxia and psychiatric features. And at the NMRI, she has a severe cerebellar atrophy and some leukoencephalopathy. And she has a history of psychomotor delay since she was 2 years old. A muscle biopsy was performed, and we found a severe COX deficiency that we can see in this staining, and in the muscle. And muscle biopsy confirmed the severe COX deficiency in muscle, but not in fibers. We go then deeper, and we see we do the Sanger sequencing of the mitochondrial DNA, and we find a mutation never described in CO1, so in a gene codifying for complex 4. That was absolutely aligned with the finding of the severe COX deficiency in muscle. Then we go into the different tissues, and we go and we see of the patients to see the amount of mutation and the percentage of heteroplasmy. And we see that was not so high in the patients, but doing the segregation, we saw that the same amount, more or less, of mutation was present in the mother and was present in the brother, both healthy. So we went again deeper, and we do a specific model for mitochondrial disease that are hybrids, in which we can express a high percentage of mutations. And we see that in hybrids with 100% of mutations, the biochemistry activity was not presenting with a severe COX deficiency. So at the end, we couldn't validate this mutation, and was not these mutations the cause of this disease. The presence of the new technologies allowed us to do the NGS. So in this case was a targeting panel for mitochondrial, for nuclear genes in encoding for mitochondrial proteins. And we did find a compound mutation in AARS2 genes that was probably the cause, the cause of the disease. So at the end, this patient has not a mitochondrial disease linked to a mitochondrial DNA mutations transmitted in a maternal inherited way, but she has a Mendelian disease, autosomical recessive, and that there is the possibility that somehow the mutations in the complex 4 genes, mitochondrial mutation in the complex 4 genes, could somehow have an impact in the effect of COX in this specific patients. But again, the presence of the 2 technologies and the combine of the 2 genome can give vision of the mutation of the disease. And the second case that I want to show you is a case of a patient, an adult patient that presented bilateral ptosis and ophthalmoparesis. So it just I can say just a little bit of muscle involvement. The muscle biopsy was done, and he has a few COX negative fibers. But in the family history, we see that the mother, the grandmother, and the 2 aunts was affected of the disease. So it looks that there was a autosomal dominant inheritance. At the muscle biopsy, we did the Southern blot, and the Southern blot showed the multiple deletions of mtDNA. So instead of going by gene, we do again a target NGS panel. for specific gene associated with PO and PO+. And we found POLG mutations. I mean, POLG is one of the genes that is involved in this replication machine that is linked with multiple deletion and that can give an external ophthalmoplegia. So the diagnosis was done. We had just one mutation, and so with the segregation we could go deeper and see if they segregate in the whole the family, but looks like the diagnosis was done. But actually with the panel, we could sequence more than one genes, and we find a second mutations, again, a hetero mutations, so just one mutation, pathogenic in the twinkle genes, that is again a genes that is associated with external ophthalmoplegia with mitochondrial DNA deletions, that is again autosomal dominant. So the idea is that maybe these two mutations can have an impact or more, and they can somehow be both part, take both part of the pathogenesis of the disease. So and this is not something that we was just the ones that find. It was published a paper where was find a degenic progressive external ophthalmoplegia linked to POLG and twinkle mutation. So with this case, I wanted to underline, first of all, the importance of this these new technologies that allows us to improve the diagnosis and open the spectrum of the diagnosis, and moreover to see that in mitochondrial disease, like in other diseases, this case of degenic mutations, so the involvement of two genes are not so infrequent, and with this new technology we are finding more and more patients in which there are two maybe heterozygous mutations that being together can give allowed the phenotype. But with this the perspective of the future. As I said before, still 50% of the undiagnosed cases of mitochondrial disease are not diagnostic, or they are present of VUS. VUS means that we have these mutations that looks like to be responsible for the disease, but we can't really prove this the impact of this mutation in the providing the phenotype. And so there are again an expanding of new technologies, and the new technologies are the transcriptomic or proteomic. So instead of analyzing only the gene, so the DNA, we can go deep and analyze the tRNA, so the transcript or the proteins, and see if there is some alteration in this two part of the machinery. So in the tRNA in the RNA with the RNA sac on the proteomics to the protein level. And like this, we can see some splice, aberrant splicing or monoallelic expression, and then go back to the genome of the genome to see if they are really pathogenetics. And this again can improve the diagnostic rate of this disease. And the diagnostic rate of this disease can be much more improved again if we took these methodologies all together, and we can have a score and amount of more diagnosis about 10 or 35%. Actually, these methods are quite expensive and are difficult to analyze at the moment. So we couldn't use this method for the normal diagnosis of our normal diagnostic work in our diagnostic lab, but for sure they can be the future to identify new genes and new mechanism of this disease. And I'm finished with my talk, and I just want to say that again, mitochondria are the powerful, they are giving our energy. So as Bart Simpson say, I tried to see how they are not important, but then I lost my energy and I was too tired. This is my group and the lab in which I work.
Thank you very much, Costanza. Very nice, elegant presentation